DNA methylation is a widely profiled epigenetic marker that impacts gene regulation, disease, and more. In addition to studying methylation from a singular species, data across multiple species is also highly informative and has the potential to impact applications such as comparative biology, studies of species evolution, and studies of aging and development. I will specifically discuss this aging application throughout the talk. Various methods exist for profiling methylation data, from sequencing-based methods to microarrays for specific regions of interest. Despite microarrays covering less of the genome, they still provide an easier and more robust data collection method, and thus are still widely used in epigenetic studies. Historically, microarrays have primarily been available for human and mice. This technical limitation has contributed to a strong emphasis on the study of methylation in humans as opposed to other species. So, with the utility of cross-species analysis, this begs the question, can we use microarrays to profile methylation in other mammals? With the development of the mammalian methylation array, we can. This array allows profiling of any mammalian species in a common set of over 37,000 methylation probes selected from highly conserved loci among mammals. Selecting from these conserved regions of the genome increases the probability that insights gained in one species can transfer to another. This array has been applied to over 300 species spanning over 50 different tissue types. This data, published in this science paper highlighted here, has been applied to studies of aging across species. Despite this data covering a wide range of species and tissue types and already showing utility in these aging studies, only 3.6% of the potential combinations have been profiled. The difficulty of acquiring samples in certain species and tissues, such as rare species or tissues that are lethal to collect, make it very difficult to experimentally profile this entire grid. Thus, our goal is to use the available data to predict DNA methylation values of all the array probes in these missing species and tissue combinations. We developed CM-impute, or cross-species methylation imputation, to perform this task. CM-impute specifically seeks to impute a species' mean methylation values within a particular tissue type, which I'll henceforth refer to as a species tissue combination mean sample, or combination mean sample for short. This type of mean methylation sample has already been used to study species-level characteristics such as maximum lifespan or gestation period. CM-impute is trained on different categories of the observed data to learn the different methylation patterns between species and tissues. In this example, I'll describe the data that could be used to impute these missing horse tissues shown in light green. The first category is same species, different tissue information, which allows CM-impute to learn the observed patterns among these known horse tissues. The next category is same tissue, different species, which allows CM-impute to learn the differing methylation levels across the mammalian species in these target tissues. The final category is the overlapping species information, which allows CM-impute to learn the cross-species methylation patterns in the overlapping tissue types between horse and the other mammalian species. All of this data combined allows CM-impute to make an informed prediction of what these missing horse tissues' methylation values could be. Each of these samples in the different categories I just described are inputted into a generative neural network framework called the Conditional Variational Autoencoder, or CVAE. Each of these input samples also come along with a one-hot encoded species and tissue label denoted by the Y in this figure. This model is trained to both encode the input samples into a latent space and then subsequently reconstruct these samples as accurately as possible. During this process, the latent space is also regularized to be as close to a standard normal distribution as possible. Through this regularization step, we create a probabilistic space that allows the CVAE to not only encode an input sample into, but also generate brand new samples from the latent space. By including the Y term during training, we allow control over the types of samples that can be generated. Thus, once trained, the labels of the target species and tissue combinations can be inputted into the decoder, which, with the learned species and tissue relationships, is able to impute a sample that represents these missing combinations. To test CM-impute, we performed a 5-fold cross-validation analysis. Each fold was randomly selected with the criteria that when a species-tissue combination is held out, there is still same species, different tissue, and same tissue, different species data available during training. Here I show an array of all of the observed held out samples with the samples and the probes in hierarchical clustering optimal leaf ordering. This shows that the samples cluster by phylogenetic order and that the probes cluster based on lower and higher methylated regions. We additionally visualize this data with the species signal removed, which reveals differentially methylated regions that are highly associated with tissue type. We then compare the CM imputed combination mean samples to the observed held out samples and see that there are similar methylation patterns. We're still clustering by phylogenetic order, seeing similar stratified patterns across different regions of the genome, and showing these differentially methylated tissue regions. We quantify this performance using the sample-wise Pearson correlation between observed and imputed combination mean samples. CM impute achieves high imputation performance with an average correlation of 0.92 across all of these samples. We additionally show the natural variability among individuals within a combination as a reference for this performance. We compare CM impute to 4 baselines, the first being the species baseline, a mean imputation-based approach that predicts the average of all training samples of the target species. Then logistic regression, which predicts the methylation values of each individual probe from its species and tissue label. The tissue baseline, which predicts the average of all training samples of the target tissue. And the global baseline, which predicts just the average of all training samples with no filtering. CM impute outperforms all of these baselines. We next investigated CM impute's probe-wise Pearson correlation between the observed and the imputed probes, in which it achieves an average correlation of 0.69, with this correlation increasing to 0.77 when considering higher variance probes as shown in this figure. CM impute once again outperforms the baselines in both of these contexts. We further dig into this probe-wise performance by evaluating how the observed variability across probes impacts said performance. We consider here how much a probe varies between different tissue types, and we specifically use mean squared error for this analysis, as Pearson correlation is less informative for lower variance probes. As you can see, CM impute remains relatively robust to lower and higher variance probes with a fairly consistent MSE across them. When we specifically look at the species baseline, which is the highest performing baseline, we see that for lower variance probes, there tends to be a low MSE. This makes sense because if a probe isn't changing across tissue types, then a mean imputation-based approach can be effective. However, as this variance increases, so does the species baseline MSE, showing it's not as robust to these mid and higher varying probes, which tend to be what are interesting to look at in epigenetic studies. We additionally show that CM impute outperforms the remaining baselines across lower and higher variance probes. Once we validated the performance, we used CM impute to impute 19,786 new species tissue combination mean samples that fill in the remaining 96.4% of the potential combinations in our grid, shown in magenta in the figure on the far right. While we don't have the observed data compared to in this context, we still see clustering by phylogenetic order and the stratification patterns across different probe regions that we would expect based on the cross-validation results. We additionally see the differentially methylated tissue regions that we would expect, showing that we're capturing both species and tissue signal in these CM imputed samples. We further dig into these species and tissue signals by looking at the pairwise correlations between combination mean samples. We'll first look at the observed data and see that the observed data yields a highly correlated block structure among samples of the same phylogenetic order, and within that, within samples of the same species. We see here that there's a very strong species signal in this observed data. The patterns among combination mean samples of the same tissue type have a less discernible correlated structure. We next look at the CM imputed species tissue combinations. While this covers a vastly different set of species and tissue combinations, so we can't directly compare to the observed, we still see these similar patterns of highly correlated block structures within phylogenetic order and within species, showing that we're still capturing similar signals to the observed, even if we can't compare directly. We quantify these species and tissue signals using the area under ROC curve to evaluate the pairwise correlation's ability to predict whether the two samples included in that pair are from the same species or the same tissue type. We restrict this analysis to the cross-validated samples in order to directly compare the observed and imputed signals. we see that CM-impute, shown in blue, yields similar AUC values to observed, shown in red, for both the species and tissue groups. None of the baselines are able to achieve this for both groups. To demonstrate the utility of our imputed combination mean samples, we conducted a leave one species out linear regression analysis to predict the logarithm of the maximum lifespan of various species based on their species level methylation data, similar to the Lee et al. Study shown here. When we use exclusively the observed data, we achieve a 0.81 correlation between the predicted log maximum lifespans and the reported maximum lifespans reported in the ANAGE database. When we repeat this analysis using exclusively the imputed data, we achieve a similar predictive performance across this same set of species. Additionally, the final predicted lifespan values using the observed data and then using the imputed data strongly correlate with one another, showing that they're not just similar in predictive performance relative to the ground truth that we define, but they're also predicting similar values. This shows that the imputed data is a valid substitute for the observed data in these types of downstream analyses. In conclusion, the development of the mammalian methylation array greatly expanded the amount of cross-species methylation data we have available, but left gaps in the species and tissues. Thus, we developed CM-impute, which uses a conditional variational autoencoder to impute the missing species and tissue combinations. CM-impute yields high imputation accuracy and maintains species and tissue characteristics that we would expect based on the observed data. And finally, the imputed combination mean samples can be applied to downstream analyses, such as predicting maximum lifespan. I'd like to acknowledge my lab, the Mammalian Methylation Consortium, and all of the funding sources. You can see a link to the preprint here, and I can take any questions.
You have a question?
Do we have time for a question?
One question.
Yeah. So when you left out the species, did you do it in a phylogenetic, was it random? Or was it like, can you say something about, I mean, you left out only one species, did you maybe do 5-fold cross-validation or something? And something on those lines, for example, can you predict something where you can predict the methylation of a species that is far away from other species?
So the question was, when we held out samples in the cross-validation analysis, if we were like informed based on the phylogeny of these species, and if we could predict like a species that's really far away phylogenetically than the stuff that's included during training. And we did select the random folds completely randomly, no information based on the phylogenetic order. I don't have any specific data on if we can predict like a sample that's like farther away phylogenetically versus closer. I do know, but I do see that we are able to predict all sorts of species. So I would be fairly confident in stating that yes, we could do that, even though we didn't, especially because we selected randomly. There must have been one that was phylogenetically far away, and we see that we achieve high performance across all of the species. There isn't like certain ones that are better than others.